Okay. Hello, everyone. Welcome to our week 7 lecture, finally back on new content. As I mentioned in class last week, I intended for this to be 2 separate lectures. I've cut down a little bit, and they were already pretty related in content, so it should be okay. But this recording might be slightly longer than usual, and I'll leave that note in my email to you guys, so that hopefully you can get a little bit of an earlier start if you are not someone who likes to sit down and watch 25-minute videos. We'll see how it goes. Okay. So this week we are talking about extremophiles. So I've kind of brushed over them a couple times in class. We're going to talk about what an extremophile is, and then we're going to talk about surviving a bunch of different types of extreme environments. So we'll talk about temperature, we'll talk about osmolarity, we'll talk about pH, and we'll talk about pressure. And in all of those instances, we'll talk about why it's difficult for organisms to live in those environments and how microbes do it. Okay, so starting off, what are extremophiles? Extremophiles are microbes that can survive in extreme environments, right? And this is what we as humans and animals define as extreme. So an example would be living under the Antarctic ice sheets in a frozen lake or at the bottom of the ocean. I've mentioned the organisms that live at the bottom of the ocean next to the vents where they're getting sulfur. So that's an example. Usually these are bacteria and archaea. Archaea specifically tend to be pretty common extremophiles, but that's not always the case. Okay. And as I mentioned, we think the conditions they're living at are extreme, but they are actually thriving. These are not organisms that are living at the very edge of their own existence. They are living happily, comfortably, and thriving. And this is, in fact, the only environment in which they are adapted to live. So this is a great example of how we might find organisms adapting on other planets that seem uninhabitable to us, but are actually, possible for life to survive in. Okay, so I want to start with temperature. Why is temperature so hard to survive at the extremes? So this is an image of fried egg. So in this, when you fry an egg, essentially what happens is that the proteins of the eggs get denatured and the molecular structure changes so that you get a completely different outcome. And then you have an image of a frozen Coke can in which the water in the Coke has frozen solid and has ruptured the organism. So we can think of these as the two extremes of hot and cold temperature, and these as analogies for what would happen to a microbe. Okay. So essentially what's happening to a microbe, cells have membranes and walls that protect the cell and regulate the entry and exit of molecules. And these are what defines an organism of this size, right? So we talked about surface area, volume, et cetera. So the cell wall, cell membrane are what are giving a microbe its shape and its function. But as temperature changes, the ability of these membranes to function also changes. So when you have increased temperature, you're increasing the fluidity of the membrane. The pieces that make up the membrane are moving past each other. The proteins can move more. The lipids can move more past each other. And you start to leak and you start to denature crucial proteins. When you decrease the temperature, the membranes do the opposite. They become stiff and they become really rigid. And they have slower reactions that can make it hard for a cell to get nutrients. And a cell can even fully freeze solid. So microbes must combat both membrane issues and metabolic issues to survive in any extreme temperature. So they must be able to maintain a shape and a structure, but also maintain this ability to bring in nutrients and put out waste. So these are temperature gradients at which different types of extremophiles live. So as I mentioned, these microbes are thriving at these temperatures, right? They're not just barely making it like we would in the desert. These are microbes that love hot conditions or super cold conditions. So each of these has a peak where that is the ideal temperature for which these species and these groups to live. So psychrophiles live at extremely cold conditions, and they are happiest around 4 degrees Celsius. Mesophiles, which are generally the ones that will spoil food, like E. coli, live around 40 degrees. Thermophiles like heat, and they live around 60 degrees. And then you get hyperthermophiles that live at 88 degrees and almost 100. So you can really have a broad range of temperatures in which microbes can live. Okay, so the next thing that I wanted to talk about was this idea of osmosis and salt concentration and how organisms can survive that. So I wanted to harken back to some 8th grade through 12th grade science and talk about osmolarity pressure. So cells are essentially just a sack full of water and other stuff in it, so like dissolved salts in water. And they are in a broader solution of salts in water. And when their environment has a higher concentration of molecules in it than they do, the cells let out water to become balanced with their environment, right? So the energetically favorable, right, in terms of physics, in terms of statistical mechanics situation would be for the environment and the cell to have equal concentration to each other. So when they are out of balance, the cell either lets out water to become balanced or takes in water. And to the extreme, this can cause irreversible rupture or shriveling of a cell, and can cause the organism basically to die because it's not in balance with its aqueous environment. So here is just a cute little GIF of where water is moving in terms of hypotonic, meaning that the cell is in a solution that has less molecules in it than it does, or isotonic, where it's equal, or hypertonic solution where the water would be exiting the cell. And hypotonic, the water is entering the cell because there's more water outside. Hypertonic, the water is exiting the cell and the cell shrivels. Okay. So this is just a question that my advisor suggested for you all. Why doesn't your pasta rot in the box? So we keep dried pasta for a really long time, just sitting on the shelf, and it doesn't go bad. My bread goes bad. Cereal sometimes gets stale, but not really in the same way, I guess. So why doesn't pasta rot? Pasta doesn't rot because it's dry, because nothing is living in it, because there's no water, right? So microbes need aqueous solution to be able to have reactions with their environment. So all microbes, even if they're living on like a surface, there is enough water for them to survive, or oils or something in which they can be in solution. So that's, if you think about COVID, right, why COVID spreads through droplets more than it does through anything else. Okay. So microbes that are surviving in these high salt concentrations, right? So anywhere where they're in a hypertonic solution and they would lose water to make themselves equal to the solution, those are called halophiles. Halo meaning salt and phile meaning loving. Halophiles adapt their membranes to be able to pump in more ions to balance out the osmotic pressure and prevent themselves from shriveling up because they've given all their water up to their environment. So basically they're taking in a bunch more salt to make it so that they are balanced and they don't have to give up all their water. So this is just a picture of a pink lake in Australia called Lake Hillier, I believe, and it has halophile microbes in it, and that's why it's pink, because they do reactions where they are pink or something like that. So pretty cool. Okay, so the next of our 4 extremophiles to examine is pH extremes. So pH is slightly more interesting than salts, but you can start from the same baseline. pH is a measure of hydrogen ion concentration in aqueous solution. So I talked about salt concentrations, where these like positive or negative ions in a solution, whereas pH is just a measure of H+ ions. And you can see in solution, water always forms a small amount of these ions And if you add other piece compounds to it, like acids, you get more H plus ions, and bases, you get more OH negative ions. pH is a logarithmic scale. Each step is actually like a 10-fold increase or decrease in concentration. If you guys are familiar with the Richter scale, so how we measure earthquakes in Los Angeles, that is also a logarithmic scale. So pH 1 versus 2 is a huge difference. Low pH tastes sour to us, and high pH tastes basic, and it's also slippery. If you think about why soap kind of feels slippery to you, which I always thought was interesting. Okay. Here are examples of things on the pH scale. Acidic is towards 0, and then neutral is 7, water. It's basically the amount that water naturally ionizes to be equal parts OH and H. And then alkaline, basic, towards the positive end. Yeah, and like I said, soap is basic, lemon juice is acidic, and you've got some things in the middle there. Okay, and then here are a couple more, another scale, but what I liked about this one was how it shows how the ion concentration converts to the pH value. So if you had an H plus ion concentration of 1, essentially, you would have a pH value of 0 and hydrochloric acid. So essentially the strongest acid that you could have. So this is just a value of the concentration of the H plus ion and how it converts over to a scale. Okay, so why is pH particularly tricky? So we know osmolarity is already a factor. So pH can cause rupture and shriveling, but H plus ions and OH ions can interact with different molecules of the cell. So they can cause damage to proteins by bonding or binding to them and adding an extra hydrogen to a molecule that didn't originally have one, or taking one away. And it can cause attractions that lead to unfolding, right? So if you have a really acidic solution and a protein that has a bunch of negative charges, the negative charges are going to be attracted to those positive charges in the water, and the protein might unfold. OH ions can also cause damage in a similar way, and they're high in concentration when you have high pH. So this is just an example of what happens to an egg when you put it in different pH concentrations. So these are increasing acidity. I believe orange juice would be more acidic than Coke. So nothing happens to egg and water. In Coke, you start to see some damage to the shell, but by the time you get to vinegar, the shell is almost completely gone, and the outside of the white and the yolk have been kind of converted into like a thin sheen where they were touching the acidic thing. So how do cells survive pH gradients? They have to, again, balance the osmotic forces, and requires energy to expel the ions and balance that force, or bring in ions like they would in a high salt concentration. They also can change the structure of their proteins by including other amino acids. So, going by. Okay, so what you're seeing is slightly different variations of the same protein. So they evolved to include different amino acids that don't have these positive or negative attractions to higher pH. So they're creating proteins that are less likely to unfold in their environment. So that is natural selection, right? So they accidentally have a mutation that gives them, a protein that's less likely to unfold, and then they become more, I guess, able to survive in that environment. So that's pretty cool. Okay, I think that's all pH. Okay, yes. The last thing I wanted to talk about is pressure. So pressure, physically speaking, is a force exerted on an area. The surface pressure on Earth is determined by the atmosphere. So the pressure that I feel on me right now is the weight of the column of air directly above me that is pushing down on the area of the top of my head, essentially. So high pressure, essentially like it can smush a human, can smush cells. Especially this is relevant in like deep sea or under ice species where they're experiencing like high amounts of weight on top of them. And it's similar to extreme cold in that it causes cells to be more rigid, right? So they are, pressure and temperature are related in a lot of ways, but extreme cold makes a cell more rigid. And so you are seeing species that need to be able to adapt to that. Low pressure could theoretically expand cells too much. And this could be relevant to airborne species on other planets, but is not necessarily relevant to things that we see on Earth. Because at the point where we're talking about the atmosphere, it's just not thin enough, right? So by the time you get to the point in the atmosphere where there's not enough water for cells, the pressure would still be okay. They wouldn't just like explode. So this could apply more to other species if you're thinking about atmospheric species. So this is an example of what happens to a styrofoam cup when you take it down into the bottom of the ocean. And this is showing the depths of how it's compressed. And I just thought that was really interesting. My advisor for this class, Tina Troyd, also does this in her submersible and has a couple pieces of compressed styrofoam in her office, which are pretty cool. Okay. How do microbes survive these extreme pressure changes? So microbes that love high pressure are called piezophiles, loving, I guess, pressure. And their membrane fluidity, so their ability of their membrane to move against itself and be able to take in and put out ions, needs to be increased to accommodate for a higher pressure because the pressure wants to make it less fluid. So they do kind of similar things as would something under extreme cold, right? Like they would increase their the compounds in their membrane, like the lipids that would make them more able to move against each other. In a thin atmosphere, like I mentioned, you would probably have to get better at capturing molecules from your environment, right? So we talk about how cells need an aqueous environment, but if they're in a really thin atmosphere, there's probably not a lot of water around them, so they need to get better at capturing molecules. Yeah, so one of the things that I also mentioned here, and this is probably not clear, I'll try to fix this on our written slides that I post. You would need to have a stable environment, right? So piezophiles are not, as I mentioned, microbes that want to go from low pressure to high pressure to normal pressure to back and forth, right? If you're altering too much the environment, you are still making a microbe that is not adapted to its niche, right? Because it's adapted to really high pressure or to really low pressure. And especially in really low pressure, if you moved it to higher pressure, you would probably find that really damaging because the cell would have been way too rigid. So essentially that's tying back to the idea that I started with, that these are microbes that are thriving in their specific environment. They are not just super flexible, always able to survive microbes. They are just well adapted to their specific unique environment. And at extremely high and low pressures, just as extremely high and low temperatures, the states of matter change. So if you put room temperature water in really low pressure, it will boil. So that is also something that needs to be taken into account for low or high pressure organisms. So this is kind of mentioned on the previous slide, but how would a microbe survive extremely low pressures? And how would it potentially adapt itself that it would not rupture, or that it could still have some shape that helps us to define it as a unique organism without being completely absorbed into its environment, or just kind of a mass of proteins that have kind of expanded. And that's something I'll just leave you guys to think about. Okay. So I can't fit everything on this slide because we tried to cram 2 weeks into one lecture. But the main points: what is an extremophile? So loves extreme environments, but also recognizing that that is a specific niche for them and they are adapted to a specific environment. And those environments include temperature, salt, pH, and pressure, the ones we've covered. Yeah, and to think about the ways in which they have to adapt to survive and what changes they have to make to their proteins, to their membranes to be able to do so. And here are your opossums for the week. I was just thinking about them being all cozy and cuddly, and I saw this guy, which I guess he must have the edited eyes to make him look extra cute, but he's a little opossum burrito, and they're so cute. Anyway, I hope you love them as much as I do at this point, because I've only got a couple more weeks to convince you. All right, I will see you all on Monday.